Hello everyone, welcome to week number 3 in Bio 120. This week is a really cool week because it's all about cells and I love that. Okay, so let's see what we've got going on. First of all, we want to remind ourselves what we learned about in week 2, and that was about atoms, elements, molecules, macromolecules. So you learn the importance of water in cells, very, very important. You learn the importance of carbon as the backbone of all organic molecules, terribly important because you can produce many complex, gigantic molecules by using carbon atoms as the backbone of your molecules. We learned about the 4 groups of macromolecules: carbohydrates, the lipids, and the proteins, along with the nucleic acids, and you had to answer some questions about those, like why aren't nucleic acids on food labels? And I hope that you figured out that's because they're made up of sugars and basically amino acids, which are the building blocks of protein. So you don't have to have nucleic acids on labels. So anyway, this week we're learning about classification to start with and cell structures. So classification is just how we as scientists put organisms into groups of like organisms. And they can be gigantic classifications like domains. There are 3 domains: eukarya, eubacteria, and archaea. Archaea contain archaebacteria, which live in very extreme environments like hot springs, like down deep in the ocean, like in ice, all those sorts of locations. Eubacteria are found everywhere else. They're the ones that live in and on us, out in the soil, out in the environment. Those are the eubacteria. So far, out of the 3 domains of life, 2 of them contain bacteria, and so that leaves eukarya for all the rest of the organisms. Eukarya contains the eukaryotes. The eukaryotes consist of eukaryotic cells. And EU means true. Kary means kernel, which refers to the nucleus in cells. So this gives you a hint as to that there may be some differences between the cells of eukaryotes and prokaryotes, which are the bacteria. Pro means before a nucleus. So there are definitely huge differences in cell structures between eukaryotes and prokaryotes. And so that's going to be important for one of our questions coming up. The chapters for you to read. Chapter 20, the first section. That's it. That's why I put 20-1 here. And then chapter 4, skim through it. That's all about the different cell structures in bacteria versus in eukaryotic cells. The major difference is that in eukaryotic cells, there are all these little compartments called organelles: mitochondria, a nucleus, the Golgi apparatus, the endoplasmic reticulum. Oh, there's lots and lots and lots more. But in bacteria, all the molecules in the cell are just in contact with each other. There's no nucleus. Bacteria aren't big enough to have a nucleus. A nucleus in a eukaryotic cell is way bigger than a bacterial cell. So there are huge differences in the cell structures. Okay, read the Module 3 overview. This gives you a good overview of the differences between prokaryotic and eukaryotic cells. It really does. Watch a few of these videos. They're very short. I'll also add a couple more that I'll put in the announcements. And please, please watch the one that I have that is the Harvard antibiotic resistance study, and how quickly antibiotic resistance can happen and how resistant the bacteria can become in an extremely short amount of time. This is a wonderful, wonderful experiment. So please go and look at it. I think the video is only 2 minutes long or something, but it's very, very cool. It's one of the best videos I've seen in a very long time having to do with bacteria. So please watch that because it will help you answer some questions. All right, and then, so the assignments this week, you've got discussion board postings, as you do almost every week in this course. And your initial post needs to include why antibiotics. So what are antibiotics? Antibiotics are drugs or chemicals that are used to kill bacteria in an organism, usually an animal. There are chemicals that are used to kill bacteria in animals, for example, and not kill your cells. And that's really important, right? Because yes, I can kill all the bacteria in you by radiating you, but that means you're going to die too. So that's not the goal of antibiotics. So why do these chemicals target bacterial cells and not our own eukaryotic cells? This is terribly important, and that's because there are differences in our cell structures, and that's why you need to understand that concept this week. And then also you're going to answer the question, how does our use of antibiotics as human beings, because we're the ones who give antibiotics, right? So how does our use of antibiotics create and facilitate and increase antibiotic resistance in bacteria? That is one of the top 10 public health problems in the world today, antibiotic resistance. So we're giving people all these antibiotics to cure bacterial infections, and then the bacteria react and evolve resistance mechanisms to these. So we're running out of antibiotics. An example of this would be MRSA, methicillin-resistant Staphylococcus aureus, which is a huge problem these days. And then in your response posts, remember, response posts are due by Sunday night, initial posts due by Thursday. Your response posts need to include whether we should be putting antibiotics in livestock feed. And I don't mean using antibiotics in livestock to cure infections and diseases in them. This is routinely put into livestock feed because it keeps the livestock healthy, because they all live together in small spaces, but also it helps increase their weight quicker than without the antibiotics and more. And the reason for this is those antibiotics fight off silent infections that you don't even know are there. All right, so that's the discussion board postings. You're also going to begin your final project. So first of all, you have to pick one. Last week, I talked about the different topics that we have listed. Go back and listen to that week 2 video at the very end if you want to hear my explanations of those, because I'm not going to do that again this week. That would be redundant. Choose your topic. If you want to choose something that's not in the list, you have to email me. You have to get it approved before I can grade it. So make sure you do that. I'm always looking for my email messages, so I'll answer you at least within the day, but usually much earlier than that. Follow the milestone 1 guidelines and the rubric. The guidelines are the instructions. The rubric is how I grade, right? Make sure you follow those because your instructions are there about what to do. Another thing, use the correct format. This is not an English essay. This is an organized outline where you are answering the questions about your topic so that you can get all your information in a good order, in the right order, so that you could explain this to someone eventually, which will be the end of your final project in week 7 of our course. So use the correct format. It is an outline with details. And I'll show you that in just a second. Make sure you label your sections. All right, let me show you an example of that. Let me get to the next page. All right, look down at the bottom. There's a list of the topics right there. Wait a minute, you can see my face. There you go. You see a list of the topics there. Then look down under that and look at that outline. So the first section is called Introduction. Okay? So say you choose GMOs. All right? So look at Introduction. Describe the topic you selected. I don't know how many students say my topic is GMOs, and then they just go on. Okay, a lot of people don't know what GMO stands for, genetically modified organism. But then you have to explain, well, what exactly is a genetically modified organism? Some people don't understand that. You're going to assume your audience doesn't know very much about your topic, right? So describe. Anytime you see describe, that means give a good description. Don't just put 3 words down. That's not a description. And then answer the question, why is this significant or interesting to you? Why do you care about GMOs? Yeah, why? Why is it significant to the field of biology? Give some real-world examples. There are all kinds of examples of GMOs, golden rice. Almost every corn plant in the United States is a GMO. Tomatoes are. Oh, there's so many crops and plants now that are GMOs that you would hardly even know. So take a look at those. All right, so that is section 1. Big Roman numeral 1, introduction. Type under that. Section 2, topic. Then there are 3 subsections there. Where? Where does this particular thing happen? Does it happen in ecosystems? Does it happen down on the cellular level? Does it happen in biomes? Where does it happen? If you're talking about GMOs and you're talking about genetically modified organisms, then that's on the species level, right? Can it affect ecosystems? GMOs? Yes, it certainly can. So you would include that. Where does it happen? How? How does this happen? So this is where you would describe how GMOs are made. How? If you're talking about another example, like water pollution, how does this happen? Well, how does water get polluted? Dump in excess fertilizers, dump in chemicals, runoff, just soil runoff from construction areas and those sorts of things. There's lots of ways that water pollution happens. So that's what you would describe. Who benefits from this? Now, sometimes if you're talking about eutrophication, algae benefit greatly from additional fertilizers that have been dumped into lakes and ponds, or the ocean even. So the algae benefit from this, and then they cause something called red tides. But also, who else benefits? And usually, if this is a problem, if we're talking about a problem over here in our list of topics, you can believe that some group of human beings is benefiting from this. So if you're talking about water pollution, who benefits from dumping chemicals into bodies of water? Well, lots of different industries, because then they don't have to clean their water that they use to cool down their industrial plants or whatever it is. They don't have to clean it. They can just dump it out there in the environment and they don't have to worry about it, right? So who benefits? Who benefits from this particular topic? So those are the things that you need to cover this week. All right? And as we go along, you'll add more sections to your final project. This is the first part of your final project. And after I grade one of these milestones, you can go in and make corrections based on my feedback. That is really important, because by the time we get to week 7, I will have graded every part of your final project. All you have to do is make your corrections, fix the parts that you need to fix, put them all together in a slide presentation, and you will have a perfect presentation. So please do that. I mean, that's to your benefit. And then finally, the tip of the week is put your assignments in the correct format. There are always formats that you have to use, whether it's an English paper or doing math or working physics problems or doing a biology assignment like ours. Ours is an outline. There is a template in the guidelines. Use the template. It gives you the outline for you. I don't know really why you need that, but anyway, it just makes you use the outline format. Okay, there are other instructions about this document, like make it double spaced. Yes, and that's for my easy reading. I do really want that. 12-point Times New Roman font. I don't care what font you use. Just make it readable, right? Make it easy on me. So just make it readable. 1-inch margin. I don't care. I'm not going to measure your margins. I will not measure your margins. Don't center justify your document, though. That drives me crazy. That makes it not easy to read. You know, just make it easy to read. You know how to do that. 1 or 2 pages. Yeah, this does not need to be a dissertation. It's not going to be 10 pages long. And if yours is 10 pages long, that is way too much. Yeah. Use in-text citations because you are getting this information from somewhere. Well, where? Don't tell me Google. Oh my gosh, no, 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 no. So where are you going to get the information from? The Shapiro Library. You got your textbook. You can use Google Scholar. I said don't use Google, didn't I? But Google Scholar has all the scientific articles that are out there. PubMed is also a good database, but the Shapiro Library is excellent. So use it. Plus your textbook. Your textbook is also excellent. Use those. In-text citations mean when you cover that information in the writing that you're doing, you need to give credit for that and then have a reference section as well. This is not all just coming out of your head. I can't imagine that you know this much about these different topics, that everything just comes out of your head. So make sure you use citations and include a reference page. All right. If you need anything, I'm here for you. I'm in the middle of moving, and that's why my office in here is very echoey because there's hardly anything in here now. I'm in the middle of moving, selling my house. But I will be checking very often, and I will answer any questions that you have. I'm here for you, m.sigmon@snhu.edu. All you have to do is let me know. All right, with that being said, I hope that you have a wonderful week number 3 this week.